Hi, welcome to your first lecture on Chapter 1 for Human Physiology. This is your lecture outline. You guys will have a chance to get used to how I lecture and the way I put together the lecture notes for you. But I want to go through some of the main topics within this first set of notes for Chapter 1 with you. And on the first slide, you'll always have an outline. We're going to go over an introduction to human physiology, the organization of the human body, and the four primary tissues. And then finally, we're going to talk about homeostasis. That's an important concept to have right out of the gate. It's going to be used throughout the semester. So having a good handle on the concept of homeostasis will be really important. And we'll go ahead and tackle that here in Chapter 1. Okay, so your study guide outline of topics, you want to ask yourself, what is human physiology? You want to be able to describe in detail the organization of the human body and be able to tell yourself with some level of description the four primary tissues, be able to recognize them and understand their function. And then here, finishing off a sentence like this where you're given a key term that's blanked out. Homeostasis can be described as a mechanism for control, and it's often controlled by a negative feedback loop. We're going to discuss what that is and be able to diagram these negative feedback loops, drawing an X and a Y axis. There's also positive feedback, which is more rare, but that's where we'll finish this first lecture on Chapter 1. Okay, let's go ahead and get started by defining what is physiology. It's the study of biological function of living organisms and how the body works to maintain life. Emphasis will be placed on working mechanisms, form, and function, with the goal as a student to understand the normal functioning of cells, tissues, and organs, and how they all work together, to understand the operation of specific organ systems, and we'll go through those. We'll often focus on cellular function in order to understand cells, tissues, and the organs involved. So, what does form and function mean? Okay, so structure is form. Okay, so the morphology or the anatomy of a certain organ or structure, depending on its purpose and not considering the mechanism. So, what is it? Not necessarily what does it do. Function gets into the mechanism, and that forms the basis for a lot of our physiological events that we can measure or observe. And we'll be doing a lot of that in lab, getting some hands-on experience. Function is always going to be dependent on the structure you're talking about. Okay, let's go ahead and skip to the organization of the body. Tissues and cells will be a main aspect, but we'll also have whole systems, and we'll need to consider the whole organism. So in this next slide here, the organ system and its hierarchy, if we consider the organization of the human body, a human being is an organism like any other living entity that you may have studied in biology. So the organism is supported by its organ systems, which are made up of organs working together to support life. Organs are made up of tissues, and tissues are made up of cells. Much of our focus in this class will be from the perspective of the living, functioning cell. Cells contain organelles. By definition, that is a eukaryote. Organelle function determines the overall activities of the cell. Even smaller than the cell and its organelles are molecules, down here at 6 o'clock, towards the bottom of this picture. We talk about a molecule of protein or a molecule of DNA, like what's given here in this example. Each of these have a particular structure and function that allow the cell to live. And if the cells are viable, then the tissues are alive and well, and organs will be healthy and functional too. So everything that has to do with life is dependent on cells. You guys will have a series of notes that go through your organ systems. I'm going to skip over these next 4 slides, but on the following 4 pages, you have slides on the integumentary system, skeletal system, muscular and nervous systems, cardiovascular, lymphatic, respiratory, digestive, and renal systems. I'm going to go ahead now while you take the time to familiarize yourselves with these important systems. I'm going to go ahead and let's look at an organ. Okay. What I want to get into with you is the 4 primary tissues. And if we look at this organ, the stomach in this case, this is an example of how an organ is made up of several different tissue types in order to function properly. On this cross section, we've removed part of the stomach, the outer layer, which would be the connective tissue and the epithelials. And we can see inside this organ, we see sheets of muscle. And then on the very inner portion, we see that there are epithelial cells that secrete mucus. And that's going to protect the inner lining of the stomach from the strong acid that's found in your stomach to digest your food. The stomach also contains nerves and a rich blood supply. And if you've ever been nervous about something, oftentimes you feel it in your stomach first. So all 4 primary tissues can be identified in a single organ. So in this section of your notes, the primary tissues are given, and you'll have specific examples of each. All tissues are made up of groups of similar specialized cells. You also have a review of the importance of cells and their general structure and function in this note set. The 3 main things you should be able to see are the nuclei, the internal fluid compartment or the cytosol, and the cell membrane. And I had some pictures taken from under the microscope so you can see the actual cells and you get a little bit of a refresher on cells. I'm not going over those now because I want to go over the primary tissues, but you do have them in your notes. So I'm trying to keep these mini lectures short and to the point, but you guys have the full lecture set of notes on Canvas under Unit 1. So let's go over the 4 primary tissues. Okay, so we have the tissues here that are made up of cells, and the organs in our body that are composed of the 4 primary tissues. They each have unique characteristics as to their structure and function, giving them specialization. The primary tissues include epithelial, connective, nervous, and muscle. We will go through each of these together now. Epithelials. They're typically found in areas that need to be provided with a barrier, and that can be either internal or external. So if you think of the epithelials that make up your skin, that's a barrier between your internal environment and the external world around you. You'll also find epithelials lining internal organs, forming a barrier. Your book has this example here for you. From left to right, you can see a couple of different structural configurations for epithelials. These are all epithelials. And if we look at this first one on the left, this is simple squamous, like what you would find on your skin. If you were to take a scraping of the inside of your cheek or your arm, you would see cells forming this barrier. They look like a thin layer. This is a sample taken from the lungs. So even as our bodies open up to the external environment, like through your mouth and trachea down into your lungs, epithelials are going to be there forming a barrier of protection. So the cells butt up next to each other, forming a thin layer, protective barrier. In the diagram in the middle, we see simple cuboidal. So the epithelials are modified. They look more squarish and they form a circle as they butt up next to each other. That large nucleus in the middle, and you see a hollow opening in the center. That center is usually containing some product. So these are glands that contain sweat or secrete oils, something like that. On the right, we've got simple columnar. So they look like a column. These cells are stretched out. They're long and skinny like a column. You can see the nucleus even looks a little bit stretched. This is a sample taken from the small intestine. So these epithelials form a barrier between the food you put in your body and your bloodstream. So they're specialized for absorption of nutrients. The next category is our most diverse category. This is connective tissue, or CT, and they're characterized by the abundance of extracellular material, fluid, or a matrix. They often look white in between, and we have a diverse category because this is made up of adipose tissue. We see blood, bone, cartilage. These are all considered connective tissue. CT can be either loose or dense. Loose CT is often fats or adipose. Dense CT is more fibrous, like collagen. That is a protein-based structure that forms like a scaffolding underneath your skin, so your skin doesn't sag and hang off of your bones. In these pictures, we have an example of bone here on the left. Looks like the rings of a tree, sort of concentric circles forming bone. In the middle is a sample from a blood smear. So you can see red blood cells and white blood cells. And on the right is adipose tissue. You don't see the nuclei easily because these cells are so swollen with the fat inside the cell. So they're mostly white inside the cellular portion. It's difficult to see the nucleus. But all of these are examples of CT because the cells are widely spaced with extracellular fluid or tissue in between each of them. And blood does qualify because you have a cellular portion and also the fluid plasma that contains a lot of proteins and all kinds of different dissolved molecules. Okay, so from the most diverse category, we're going to now look at the most specialized. This is nervous tissue. This is a single neuron stained purple, so it's easier to see the structures. We're going to spend all of Unit 2 on the nervous system. So this is a single neuron, and he looks structurally very different from probably any cell you've ever seen before. He looks like his hair is on fire. You can still see the nucleus in the middle, but these long extensions coming off of the cell body or soma. And then down here, as you go towards 6 o'clock, there's this long wire-like structure called the axon coming off in one direction. So these cells are very highly specialized because they carry electrical communication throughout the body. And your brain has trillions of these cell types that are very specialized for communication. And we will talk more about cell structure and function in the nervous system in Unit 2, but this is one of the 4 primary tissues. Last but not least is muscle. There are 3 different types of muscle. We will spend most of our time familiarizing ourselves with muscle using the skeletal muscle as a model for structure and function. And then we'll have a mini cardiac unit within Unit 3. The cardiac muscle is a little more specialized and has certain characteristics that you won't find in any other muscle tissue type. And then we'll have smooth muscle, which is normally found in the GI, urogenital tracts. Usually internal organs will have smooth muscle. So skeletal muscle is going to be involved in voluntary um, locomotion type movement. Skeletal muscle is going to change the angle of your bones, and your brain sends a voluntary message, an electrical message to contract or relax these muscles. The cardiac muscle, you don't have voluntary control over. It beats by itself. And so there are some structural features and electrical features that are very unique to the heart. So we'll look at these individually. And I believe you have some examples in your notes that will show you what each of these three muscle types look like. So I'd like you to be familiar with each and notice some of the differences, be able to identify them and describe a little bit about their function. And our final topic today, I'm going to go ahead and jump into homeostasis with you. I really just want to spend time on the highlights, try to shorten the lecture part because you guys do have access to my notes and the reading assignments. I want to try to tackle particular topics that need more discussion. But make sure that you look at the microscopic views of different tissue types and the cell review if it's been a while since you've been in a biology class. Okay, we come to our concept of homeostasis. This is going to be referred to over and over again throughout our course in physiology. Homeostasis, as mentioned to you in your list of learning goals for Chapter 1, it's a mechanism for control over bodily function. So you can see why this is introduced now, and it's going to be recurring throughout our course as we move through the different body systems. This is an important one to understand now so that we can apply it to the different systems that we come across throughout the semester. So organ systems maintain a constant internal environment, and you see in this example here, going for a jog in the Sahara Desert will probably raise your body temperature. And next to the picture, I have a list of different systems that are under homeostatic control. So they're regulated through homeostasis, this control mechanism I want to describe for you. So things like blood pressure, the amount of blood flow to an organ, your rate of breathing or respiration, blood pH, which is a very narrow range for normal. You can get into acidosis and alkalosis and get yourself in the hospital if your blood pH isn't tightly regulated. Body temperature, that's one we'll use quite a bit as an example for homeostasis. In fact, laboratory number 1 will be homeostasis body temperature, and you guys will do that at home. So hopefully everyone has a thermometer. Also, you see urine concentration and hormone concentrations and secretions. Those are all tightly regulated through homeostasis. And how we gain control for homeostasis to regulate our internal environment is through a process called negative feedback. These negative feedback control systems are going to make adjustments in order to lessen the impact of major external disturbances or changes, like going for a jog in the desert. Certain things that cause a disruption to the body's normal state of dynamic equilibrium, which you see here drawn in this pink ribbon diagram, there's always fluctuation. So dynamic equilibrium can be disturbed due to outside stimulus causing slight changes in the body's internal balance. If we consider temperature, if we have a set point of normal body temperature of 37 degrees Celsius, we have a flat line, okay, 37 degrees. But sometimes we get warmer than normal, 37, and sometimes we can be cooler than normal, 37. And you see the little negative sign. So if we get too hot, we want to shut that down and cool the body. If we get too cold, we want to shut that process down and warm the body. So the physiological responses would be if our temperature increases, we're going to perspire, and we can cool the surface of the skin through a process called convection. And if we get too cold, if we dip below the set point average here, our flat line, we can start to shiver. So when our muscles contract, they generate heat, and that will warm us back up. So it's this constant process of dynamic equilibrium. Balance, but in response to disruption or changes due to the environment. So the regulated variable in this case is temperature, and it's going to be controlled by returning it back to 37 degrees Celsius. And there's three parts that need to be discussed for control. We have to have a sensor. So we have to sense these changes. Are we too hot? Are we too cold? So any deviation from physiological set point that alters our internal environment, we have to have a sensor. You have sensors all over your body. We'll discuss these in detail in Unit 2 when we get into the nervous system, but something has to tell the brain or the integration center what's happening in the body. So the integration center receives this information, processes it, and determines the response in order to fix it. So this is usually the brain. The next piece is the effector organs. So once the brain issues a response, the nervous signal is going to go out to that effector organ to effect change. Okay, so effector organs produce the response that's going to restore this factor back to normal levels. Okay, so with those three pieces, the sensor, the brain, and the effectors, and for us, effector organs are going to be muscles and glands. Those are the things that are going to respond to produce change. We can gain homeostatic control, and this diagram in figure 6 from chapter 1 shows you an oversimplified regulatory response if you dip too low or if you go above your set point. The body will make changes to correct it back to normal. So you can go too low or be too high, and the body wants you to be just right, hovering around this set point. So normally the x-axis, the dependent variable for us, 99% of the time is going to be time on the x-axis. The independent variables are often going to be rate or concentration. So you see on the y-axis, miles per hour, that's rate, that's your velocity, okay? Blood glucose was given as an example here in item D. That's in milligrams per deciliter, which is how you measure blood glucose levels. That's a concentration. That's the independent variable on the y-axis. So we will do a separate video on how to draw these graphs. Normally I'd be doing these for you on the board and you would have them in your notes. So I need to get a different camera angle to show you what I'm drawing. So I'll get that set up and posted for you. But the homeostasis, if we go back to our example from our runner where he's controlling body temperature either through perspiration or shivering, those two events are antagonistic to each other, right? You wouldn't sweat and shiver at the same time. They're opposite, so they're antagonistic to each other. We'll use that term pretty often throughout this class. Let me go ahead and show you one last slide. There are some cases where negative feedback control is not being used, and it's very rare in the human body. There's only a couple of legitimate examples for this alternate mechanism. And this is when the regulation of a certain system does not return to normal. Okay, so this is rare. Positive feedback mechanisms tend to amplify the changes, and it creates a lot of instability in the body, which is why we rarely see it. But the two examples I'm going to give here are related to female reproductive hormones, estrogen that builds throughout the female monthly cycle. It goes greater, 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 higher and higher and higher. It doesn't return to normal until after the menstrual cycle. So you have ovulation at the peak of estrogen, and the same is true for labor and delivery. Oxytocin is another hormone. We'll cover both of these a little bit better in Chapter 6, which will be Unit 3. Oxytocin is going to cause contractions of the smooth muscle in the uterus to build and build and build. Sometimes it's over a 24 to 40-hour period until the baby is born. So the graph over time would look like a linear or logarithmic change. So I'm going to draw those for you when you can see what my hand is doing in a separate video. But the other example for positive feedback would be when you're bleeding. If you have an injury that causes you to bleed, eventually your blood is going to clot. It's going to then form a plug, and then eventually a scab, and then maybe in a month you'll have new skin developed in that area that was injured. That's another great example of positive feedback control. Again, that is rare. So I wanted to introduce these key concepts here in Chapter 1. You guys have access to the first 5 chapters that are required for Unit 1, and I'm going to be back with another video for you here in just a minute.